British Heart Foundation funds research into heart and circulatory diseases. One research project currently taking place at the University of Bristol is investigating cardiac arrhythmia. Cardiac arrhythmia is a prevalent underlying risk factor for cardiovascular disease. An estimated 1.6 million people are living with the disorder in the UK. The heart is the organ that pumps blood to the entire body. Every heartbeat arises from an electrical impulse. The cells which form cardiac tissue are called cardiomyocytes. In cardiomyocytes we find DNA which encodes the genes which regulate cardiac rhythm. A mutation is an alteration in the DNA sequence that makes up a gene. Some mutations affect the normal function of a gene which can impact our health. If the heart is not functioning properly, the circulatory system becomes compromised. The electrical impulses can malfunction, leading to arrhythmia. In some cases, this can lead to sudden cardiac death. Novel cardiovascular research is required to discover and test treatments to improve patient outcomes with arrhythmia. In order to research arrhythmia, a model is needed because as researchers, it would not be feasible nor ethical to conduct our research in humans. The human heart and the zebrafish heart have many similarities. The ion channels responsible for the heartbeat are also very similar to the human ion channels, making the fish suitable for cardiovascular disease research. The purpose is to induce the same cardiac genetic sequence in zebrafish to study the effect of the genetic mutation. This is performed by using a method called CRISPR-Cas9. This method relies on collecting eggs from zebrafish. When the eggs are collected, the cells are injected, facilitating insertion of genes. This method makes it possible to insert a section in the DNA that holds the cardiac mutation. Through natural cell division, the mutation is carried to the dividing cells which develops into an adult zebrafish. If the insertion of the desired gene mutation is successful, the fish will be carrying the mutation which facilitates research into cardiac arrhythmias. We hope that this research can help develop treatments for cardiac arrhythmias and help reduce the burden of this important cardiovascular disorder.